So I think I'll go ahead and introduce today's IE2 seminar speaker, Alejandro. Alejandro joined my lab as a postdoc just a couple months ago, at the beginning of September. And impressively, a week after he arrived, like literally a week after he arrived, we headed out to do field work off the Kona coast. So we were doing some blue water diving. So he somehow managed to get himself moved in and unpacked, and also get his science diving certification sorted out, which is no minor task. And also begin to learn a new instrument. He was doing some respiration measurements while we were there. So he did all of that and then jumped on a plane, although going off the Kona coast is not such a bad place to start off a postdoc. Let's see, Alejandro did his undergraduate work in Valencia, his native Valencia, Spain. And then just before coming here, he completed his PhD at Yale University in Casey Dunn's lab, where he was working on the ecology and evolution of siphonophores, this really cool, beautiful group of organisms that he'll tell you more about, I know, today. This is a really widespread and interesting group of organisms that's distributed in the open ocean and deep sea. And I really appreciate how he brings together natural history, ecology, and evolution to shed light on how marine organisms function in the ocean. And I'll let you take it away, Alejandro.
Thank you so much, Kelly, for such a kind introduction. And also thank you all for being here. As today, I'm excited to share some of my dissertation work with you all, which is going to be composed mainly of 4 interconnected chapters that are all focused on siphonophore evolutionary ecology. And then at the end, I'll give you a little glimpse into the research that I've been currently doing in Kelly's lab here at UO, a bit of what she's been talking about too. So to start with, one of the most pressing challenges in biological oceanography is to be able to use community composition alone to predict interactions between species and also to predict things like food web structure. Ecomorphology studies how organismal traits can drive ecological interactions. And then comparative biology has developed toolkits to reconstruct the evolutionary history of traits, so we can predict the role of a species based on its phylogenetic position. So the gist is, if you know evolutionary history, we can predict traits, we could potentially predict interactions. But however, there's 2 confounding obstacles that typically prevent biologists from investigating this pathway. At an evolutionary level or environmental level, local environmental heterogeneity introduces confounding adaptations for species that live in each different microhabitat that are many times unrelated to their differences in interspecific interactions. At a more organismal level, we often find what I call functional pleiotropy. That is when traits that are involved in ecological interactions are also under selection for other functions that they carry out. So in order to investigate these relationships, I had to find a place where small-scale environmental complexity was minimal, and that it's a place like the open ocean midwater, and also a system that had functional traits that are exclusively used for interactions with other species, interactions such as prey capture. And this leads us to siphonophores. Siphonophores form long chains of conjoined specialized bodies that work together as a single animal. And their tentacles carry venomous stinging cells called nematocysts that are packed into batteries. These batteries are loaded on the side branches of the tentacle called Tentilla. Just like the ammunition of a combat plane, Tentilla bear a few rocket caliber armor-piercing nematocysts and a belt of hundreds of bullet caliber nematocysts. So when an animal like this crustacean becomes entangled in the adhesive terminal filament, things start getting pretty wild as the nematocyst battery rapidly unwinds, slinging forward, wrap around the prey, hundreds of nematocysts fire on it at the same time. And then when a capture happens, the siphonophore reels up the tentacle and brings the paralyzed prey towards the mouth. These tentilla come in many different shapes and sizes across the diversity of siphonophore species, with different nematocysts adapting to capturing different prey. Some specialized for fish, others for crustaceans. But many of these elusive and understudied species, we just don't know what prey types their intriguing tentilla designs are targeting. So in summary, siphonophores are an ideal system because they live in this natural laboratory to study the evolution of ecological interactions. And they present a conspicuous amount of trait variation in their tentilla, which they use exclusively to capture prey. Another reason to focus on siphonophores, here the ones highlighted in green, is that they play a clearly central role in the open ocean midwater food web. However, it is unknown how all these trophic links are partitioned among the siphonophore species level diversity. And so are the evolutionary mechanisms that build up the complexity of this food web. My dissertation work was part of a bigger collaborative project named SipWeb, focused on studying the effects of predator traits on the structure of oceanic food webs. And my talk today is going to focus on two main questions. The first one is going to be, how do organismal traits in evolutionary history relate to siphonophore diets? And the second one is, what's the role of siphonophore diversity in midwater food webs? These two questions will be then subdivided into 4 chapters. The first 2 will address the evolution of siphonophore tentilla, contextualized with published knowledge on their feeding habits. And then the last 2 will refine our knowledge on the diets and feeding specializations of siphonophores. Siphonophores are among the largest and most abundant predators in the open ocean, with many different species for which we know almost nothing about their natural history. And while siphonophores are an ideal system for my questions, they are extremely fragile creatures, and many of them live hundreds of meters deep. So for this reason, they're extremely challenging to collect and study. So in order to get intact specimens, we need to go out into the ocean and collect them alive. And this is how I've collected siphonophores in the field. For shallow water species, we use blue water diving in the open ocean, where the seafloor lies thousands of meters below. And here we collect live siphonophores using jars. In order to collect deep sea species, we use ROVs, which are these large remotely operated submarines with cameras to observe deep sea animals that use cylinders to collect intact live siphonophores. Yep, just like that. Then this conceptual framework is going to show the propagation of heritable variants. On the left side, species evolve to inhabit different regions of the water column and therefore are exposed to different prey. Then species can evolve mechanisms to constrain their distributions to depths where better prey or more suitable prey is available. On the right side, species evolve different forms of tentilla for prey capture that are effective at capturing different prey types. Then natural selection can optimize tentilla morphology to specialize on the most rewarding prey. In combination, I hypothesized that these processes would lead to phylogenetically structured diets. In chapter 1, I explore the evolutionary history of known siphonophore specializations and their relationship with tentilla morphology. This paper was published earlier this year in PNAS. To do this, I compiled dietary data from several published sources, including gut content analysis and ROV video observations. I categorized them into distinct feeding guilds, and then also built a molecular phylogeny using ribosomal genes and a transcriptome-based backbone, and reconstructed the evolutionary history of these specializations or feeding guilds. The theoretical expectation, or the most general one, is to find specialists evolving from a generalist ancestor and then becoming unable to evolve into anything else, mostly because of the constraints that are typically associated with extreme adaptations to a singular prey type. What I found, however, is quite different, realizing that siphonophore specialists appear to have evolved from ancestral specialists on different prey, and that generalists, the ones in green here, are derived twice from specialists independently. The fact that siphonophores can easily evolve from one predatory specialization into another or into generalism seems a bit counterintuitive. And that's because most predators, in the seminal papers at least, are like vertebrates, with a prey capture apparatus that is integrated into the body. For example, the mouth of a fish is built into the skull and is used for ventilation and sometimes for territorial behavior as well as feeding, the functional pleiotropy I was talking about, right? So changing the shape of the mouth requires changes in the whole musculature system for feeding, breathing, and locomotion. And therefore, species cannot simply evolve new morphologies for prey capture in isolation. But on the other hand, siphonophore tentilla are spatially, developmentally, and functionally decoupled from all other anatomical structures, and therefore can evolve freely. So the take home here is that studying unfamiliar and weird invertebrates can actually change your perspectives on fundamental ideas in evolutionary biology. I extensively imaged and characterized the morphology of Siphonophore Tentilla using complementary microscopy approaches. So for example, I use confocal microscopy to obtain a 3D overview, and then differential interference contrast to examine nematocysts. From these images, I took 30 different measurements and ratios of, for example, lengths and widths for Tentilla and nematocysts. I measured all the different types of nematocysts that can be found in Tentilla, such as the piercing heteronemes and haplonemes, and also the adhesive desmonemes and rhopaonemes in the terminal filament. Among these measurements, here's just a curiosity, was the volume of the stenotyl nematocyst, which, as far as my record checking has gone, constitutes the largest nematocyst, not only that, the largest intracellular organelle of all living things. I might be wrong, someone correct me, please. Using these morphological data, I wanted to find out what patterns of Tentilla evolution were associated to shifts in dietary specializations. So particularly if there were any associated morphological states, distinct evolutionary optima, and shifts in evolutionary correlations between characters. Comparing the evolutionary histories of Tentillum characters and diet, I found morphological states that are associated to independently evolved specializations. An example here, we can see that both fish specialist lineages in red have independently evolved more rounded nematocysts. But I wanted to assess whether the adaptive optima of morphological evolution actually shifted with diet. So that's a different analysis. So for every morphological character, I fitted a null model of neutral divergent evolution and then compared this to a selection-based model with optima that will be allowed to shift between diets. And in fact, several characters, here's a few examples, fitted the diet-driven model significantly better than the neutral model. These traits have signatures of natural selection driving their evolution towards different adaptive states that correspond to each feeding guild. Here you can see a matrix of morphological characters on the X and the Y, with color representing the direction and strength of correlations between the rates of evolution. So for the whole tree, we can see that the rates of evolution of these Tentillum and nematocyst characters are highly correlated between them. But the question is, are these correlations constant across the phylogeny, or do they depend on diet? Do they change with diet? So to answer this, I compare the whole tree matrix to matrices within specific dietary regimes. For example, here's the matrix fitted for regions of the tree that are occupied by fish specialists. When I subtract the fish specialist matrix from the whole tree matrix, the differences appear highlighted as increases or decreases in correlation coefficients, showing which pairs of traits have dependencies that are unique to this specialization. What's even more interesting is when we compare the fish specialists to their ancestors, in this case, the large crustacean specialists. These differences are even more striking. And this is when we see strong shifts in the relationships between nematocyst-shaped characters like lengths and widths. And these correspond to these blue dots that account for a lot of those shape changes. So we have seen that Siphonophore generalists can evolve from specialists, and with these shifts in specialization, we get morphological evolution, changes in adaptive optima, and changes in the correlations between characters. These results from chapter 1 have shown that the realized trophic niche has a strong influence on the evolution of Tentilla morphology. And this elicited many more questions on how Tentilla evolved, leading to chapter 2 on the evolutionary processes leading to the extant diversity of Tentilla morphologies, now published at the Journal of Integrative Organismal Biology. We have seen that different Siphonophore species have very different Tentilla morphologies. Some are coiled, others are straight, some are enclosed in a membrane. We call it an involucrum, it's the stuff here in blue. Some also have fluorescent and bioluminescent lures to attract prey. And it's not just their shapes that are diverse, but Tentillum sizes themselves can range all the way from 40 microns to 8 centimeters. So one of the primary goals of this chapter was to provide a descriptive phylogenetic roadmap of the major novelties, such as changes in nematocyst types, nidoband shapes, and primary terminal lure modifications. When we summarize this morphological variation in a PCA, we see that much of it is explained by Tentillum and nematocyst size, and that these principal components have strongly conserved phylogenetic signals, and also indicate a relatively low morphospace dimensionality. And this low dimensionality can be a consequence of something called phenotypic integration. That is when different parts of a complex structure maintain evolutionary correlations. So I wanted to test for phenotypic integration, and I did that by examining multivariate phylogenetic correlations, not between individual characters, but between character modules, including different nematocyst types and general Tentillum shape attributes as modules. And I found significant evolutionary correlations among them, including between Tentilla and nematocysts. And this may seem trivial, but it's pretty surprising if we account for how Tentilla are built. So Tentilla are budding at the very base of the tentacle, and they develop as tentacle growth pushes them further away from the base. Like other cnidarians, Siphonophore Tentilla subdue their prey using nematocysts, which need to be assembled and loaded. At the base of the feeding bodies, Siphonophores produce specialized cells that carry nematocysts, and these are called cnidocytes. Developing cnidocytes travel down the tentacle, building the nematocysts inside the cell, changing shape and size as they go. And once they reach the tentillum, they sort themselves by type, they squeeze into this epidermal layer, they settle into their predetermined positions and rotate to point that harpoon outwards. Once everything's loaded, the outer layer then twists around the central axis into its final conformation. So the developments of Tentilla and their nematocysts are actually spatially independent, carried out by completely different types of cells. And this makes finding phenotypic integration between them exceptionally interesting, as this means that they could be likely correlated due to functional reasons maintained by natural selection. And this actually makes a lot of sense because Tentilla and their nematocysts have to coordinate together, like clockwork, to deliver a precise strike in just a few milliseconds. In fact, nematocyst discharge is the fastest accelerative motion among all biological phenomena. So funded by Yale, I set out to record high-speed footage of nidoband and nematocyst discharge across several species, and investigated how changes in the morphology correlate with changes in kinematic performance. I couldn't get a lot of data because this is very difficult to get and record live on a boat, but here's what I found. On the top side here, you can see the Tentillum discharge of 3 different Siphonophore species. I chose just to exemplify, with a corresponding static confocal image underneath. These are ordered by coiledness, increasing left to right. And I found that coiled Tentilla had relatively faster strikes. And this hints at a potential tension release mechanisms that could be associated with the coiling they undergo during development, kind of like a spring or something. And then the ones with straighter Tentilla actually had nematocysts with significantly faster shooting speeds. The filaments were faster than the ones with coiled Tentilla. So perhaps they're compensating for a lack of speed at the Tentillum level. These videos also revealed unique dynamic features of some species. For example, the spiral discharge behavior in Phrylagalmus nematocysts. And these might be drilling themselves into prey like a corkscrew, also creating an anchor. And we have no idea what these guys eat, by the way. In chapter 1, we saw that haplonym nematocyst shape has become wider in fish specialists. This is just an example of a character. But the question is, are these morphological shifts convergent? So to test this idea, I fit a multi-optimum model that also evaluates which evolutionary regimes are the same and convergent. The colors of the branches are indicating what evolutionary regime they belong to. This graph on the right is showing haplonym length and width, XY axis for extant species as small circles, and it's also showing the position of the regime optimum, the estimated optimum, as a large circle. I found that both fish specialist clades, here are the ones in cyan in this case, have convergently evolved wider nematocysts. And also this analysis revealed that the small crustacean specialists here in yellow had convergently evolved extremely narrow nematocysts at the very end, at the other end of the spectrum of shape. One last deliverable from this paper is this discriminant analysis of principal components that I made using Tentilla morphology to predict or generate hypotheses about the diets of all these understudied species that we have no idea what they eat. And then my last chapter will set out to test some of these predictions, see if we can validate what morphology can tell us. So in conclusion, I found that Tentillum characters are phenotypically integrated, that Tentillum shape and size determines discharge dynamics to some extent, and that Tentillum adaptations have evolved convergently. Going back to the two big questions, we saw that Tentilla and nematocyst traits evolve adaptively and convergently, driven by dietary specializations, evading typical constraints thanks to their modularity overall as a system. But what about the roles of siphonophore diversity in the midwater food web? Well, there's some studies that are based on visual methods, the ones that I used to do the work before, which have reported that some species are highly specialized and selective. However, it was unknown whether this pattern would extend to the deep sea and whether there are prey types beyond the detection limits of visual methods. And this takes us to the second part of this talk, starting with chapter 3, which expands the known diets of midwater siphonophores from ROV observations and disentangles the relative importance of differential prey encounter and prey selectivity in determining their diets. The main questions tackled by this chapter are whether midwater siphonophores are selective predators, whether it is their prey selectivity or differences in their vertical habitat use that drives differences in specialization, and whether they display competitive niche segregation. But how can we obtain information on feeding and selectivity for such elusive animals? Well, ROVs record hours of underwater footage at depth, for which siphonophore prey interactions, such as these we can see here, can be annotated and identified. The Monterey Bay Aquarium Research Institute has more than 3 decades of annotated footage of deep sea fauna, and this includes reported interactions of feeding and also environmental metadata, like depth, time, and other oceanographic parameters. So to the right, we can see a couple examples of vertical distributions of a siphonophore and a potential prey that were derived from these annotations. Now, from this metadata I mentioned, we were able to model the species distributions in space and time for all siphonophores and all their potential prey. Then, we compared the model distribution for each siphonophore and every prey taxon, integrating the product of each taxon's abundance across their overlap in space and time. Let me explain a bit better. For example, if you look at this graph at the top right, you can see the distribution of bristlemouth fishes in green, and also the distribution of this one large siphonophore species in purple. You can see the intersection area shaded in blue, that this area represents where feeding events can happen. So the size of it is proportional to the chance of encounter between these two taxa. And this way we were able to estimate the expected relative encounter rates between each taxa. And this constitutes a novel approach to estimate relative prey availability for midwater planktonic predators. To estimate selectivity, I then took the difference between the relative proportion of each prey type consumed and the relative proportion of each prey type encountered, estimated from these overlap ranges. On the left side, we can see the number of feeding interactions between each siphonophore species and prey taxon. These were then compared to the estimated relative encounter rates here in the center. And from these two things, we can calculate selectivity on the right, which goes from blue, meaning extreme exclusion of that prey, to beige, which means extreme enrichment for that prey. We found that indeed most deep sea siphonophores are highly specialized and strongly selective. Some species are specialized to feed primarily on krill, for which they are strongly selective. Some of the large deep dwelling species target mainly soft-bodied prey, like fishes or squids, which they also positively select for, and they exclude other prey types that are far more abundant in their environment. Apolemnias appear to have this predilection for jellyfish prey. Here you can see one engulfing a hydromedusa. However, they were not strongly selective for or against them, which points towards a more generalist role in the food web. These differences in the degree of specialization on the x-axis here appear to be driven by variation in prey type selectivity, or in other words, driven by intrinsic biological differences between the species, rather than differences in prey availability at each depth range. In fact, when I compared the relative specialization, again in the x-axis of siphonophore species, to their depth distribution, I found no relationship with their average depth. Also not with the breadth of the vertical habitat. It's not like the ones who are spread wider in the water column eat a broader variety of things. There's very little non-significant relationship. When species occupy the same spaces at the same times and they're eating the same food, they typically displace each other, forcing their distributions or diets to become narrower and less overlapping. And this is known as competitive niche segregation. So I wonder whether differences in siphonophore diets and vertical distributions that we observe could be driven by this phenomenon. So I was inquiring whether pairs of species that have high spatiotemporal overlap would have low dietary overlap and vice versa, like a negative relationship, basically. I found that species indiscriminately overlap with one another in space and time, but that is generally rare to find pairs of species with high dietary overlap, the top two quadrants. And this may suggest that competition for prey is not specific to particular depth ranges, but perhaps to the whole water column. The cases with high spatiotemporal and dietary overlap all correspond to the krill specialists. And maybe the overabundance of krill could be preventing competition from happening. Or perhaps they're targeting different krill species, but that would be beyond the resolution of ROV images. Some take-homes from this chapter are that midwater siphonophores are highly selective and specialized, that specialization is tightly linked to differences in selectivity and not to differences in depth or depth range occupied, and that there's no relationship between spatiotemporal and trophic overlap, particularly in the case of krill eaters who overlap heavily on both dimensions. They just don't seem to care. This chapter shows that the right side of this diagram has a much stronger influence on diet than the left side. And this reinforces the importance of the conclusions that we drew from the first two chapters. But in order to get a good comprehensive picture of siphonophore diets, I used molecular tools to identify prey in the gut contents of a variety of shallow and deep sea species. So to back up a little bit, there are several ways that we can learn about what animals like siphonophores eat. The most direct way would be to watch them capturing and eating prey, like in the previous chapter. And we're able to do this using ROVs, but this method is biased towards large and conspicuous prey items. Another way is to collect them and then immediately look at their stomachs under a microscope before the prey is digested. This can be done with shallow water species because they can be processed on the boat very soon after collecting. And with this method, we can see smaller things, but gelatinous animals are rarely identifiable because they're rapidly digested. An alternative to these two is that we can sequence the DNA of the gut contents to identify the prey within. Many of you know this approach. It's called DNA metabarcoding, which can detect prey that has been already thoroughly digested, which is usually the case in many siphonophores because they are infrequent ambush feeders, but also and especially in those the deep sea ones that come back from a very long, up to 12-hour ROV dive. The fact that each visual method has only been applied on a different vertical habitat has created this confound between the environmental factors that are unique to these habitats and the methodological biases. And that's when new technologies like metabarcoding can be a useful complement, is to bridge across all these methods and habitats. When prey is digested, their genomes get broken into short fragments. But these fragments can last still a long time in the guts, and at least much longer than the visually identifiable remains. So doing this work feels a bit like being a crime scene investigator, identifying the victims of prey capture. But back to this, these fragments are very short, and they can also come from very phylogenetically distant animals, fishes, jellyfish, worms, crustaceans, right? So I required small barcode regions within the 18S gene that could survive this fragmentation process with priming sites that are universal to all animals. So I went and designed and utilized 6 different barcodes within the 18S gene. And this multiplicity of barcodes help control for amplification biases, as well as providing this composite taxonomic resolution, since different gene regions have higher specificity to identify taxa for different broad prey groups. Here you can see a semi-digested mysid shrimp inside of a feeding body. And in this video, we can see that the nutrients, as well as the DNA from the prey, are rapidly being shared across the communal digestive system of the siphonophore colony. And this is why when I sampled, I always pull together multiple feeding bodies without fear of contamination, because multiple prey captures from the same colony will always become mixed up. In order to assign the sequencing reads to taxa, I needed a reference library that had good representation of open ocean deep sea invertebrates. And that is not at all the case in most public databases, because these are so hard to find and sample, right? So in my cruises, I also collected a bunch of local underrepresented potential prey taxa and sequenced them to complement these public repositories. These prey reference sequences were then backed up with photographs taken in the field of the fresh animals, and also with fixed voucher specimens in the museum for accurate identification. To do the metabarcoding, right, we first have to extract DNA from the gut contents, as well as from these reference individuals from the zooplankton. Then we amplified barcode regions using my custom universal primers, which then we sequence these barcode regions. And then finally, we can assign the sequenced gut reads to specific reference taxa in our database. Using these 6 different barcodes for the study turned out to be quite useful, as every individual barcode failed to detect at least some of the prey items. And this is because different taxonomic groups have different mutations, making their priming sites, even though they're in silico universal, they're still less likely to bind during PCR, so like false negatives can occur. I identified prey items in 47 specimens from 24 Siphonophore species. Now I want to take a closer look at just a few of them. The Portuguese man-o-war is the only surface-floating Siphonophore, and it is known to eat primarily fish, which they can ingest using multiple feeding bodies that cooperate to form like a common digestive chamber. And what you're seeing here in this video, each one of those anchovies is actually being engulfed by 2 or 3 feeding bodies at the same time. So the gate limitation, they really don't care. So my metabarcoding results found fish prey in all 3 specimens. And I also found a ctenophore DNA in one of them. And this could constitute a novel gelatinous prey type for the species. Apolemias are colossal siphonophores, and they are known to feed on a very broad variety of prey, especially gelatinous animals. Here's a recent viral video showing a spiraling apolemia more than 100 feet long. My metabarcoding detected both crustaceans and gelatinous prey, including larvaceans and salps in their guts. These prey are just 1 trophic level away from the phyto and bacterioplankton that they filter. And this suggests that apolemias in the food web could have a very proximate role in modulating carbon export and the microbial loop that these prey are involved in. Multiple Bergmania species coexist in the same deep waters and are known to eat large crustaceans. To the left, you can see a Bergmania siphonophore capturing 2 krill shrimps. Our results show that these 3 different species might be targeting different crustaceans. Crustaceans in agreement with the morphological predictions from chapter 2 for a couple of those species that didn't have any previous data. And apparently, Bergmania lata is also able to consume gelatinous ctenophores as prey. ROVs often find these Lichnagalma siphonophores eating decapod shrimps, mostly suggested shrimps. If you look closely in this picture, you can see the antennae and the eye of a shrimp inside of a feeding body. My forensic work provided a molecular confirmation of these observations, but I also found krill prey, which indicates a broader specialization on shrimp-like animals beyond decapods. Deep sea Nanomia siphonophores, like this one, are known to be primarily specialized on krill. And our data confirmed this interaction, but showed a broader large crustacean diet. On the other hand, my shallow Nanomia samples were all consuming copepod prey, like this one, which was visible through the stomach walls. This copepod specialization was notable for the Atlantic Nanomia, who were strongly selective for copepods in cladoceran-dominated waters. The siphonophore Voctius aurata, here at the bottom left, is the closest deep sea relative of the shallow water siphonophore Hypopodius, here on the top left. Hypopodius is the only described ostracot specialist among siphonophores. However, the Tentilla morphology analysis in chapter 2 predicted Voctia should be a generalist. Now, I found DNA of an ostracod, yay, but also surprisingly a bivalve in their gut contents. And now this result was pretty exciting because bivalve larvae are morphologically very similar to ostracods. They both have these two hard valves that are enclosing their body. In addition, I found a shrimp DNA and the gut contents, altogether confirming a generalist prediction, or at least validating. Spheronectes colliceri is this tiny, shallow water siphonophore, and it's a very well-known copepod specialist. This is a type of small crustacean. And we found copepod DNA in a specimen, but also DNA from a crab larva, indicating that the specialization is probably on small crustaceans more broadly. These Spheronectes also come in an acute deep sea version, and we had no prior information and no idea on the diet. We caught one digesting a small jellyfish, and this might be revealing new hidden links between deep sea siphonophores and other jellyfish. It's not just the big ones that eat jellyfish, even the small ones eat little jellyfish. I found that really interesting. And finally, we have Mugia, another tiny siphonophore, shallow water, is shaped like a rocket ship, and it's reported in the literature as being a strict copepod specialist. But I found a specimen who was feeding on a soft-bodied larvation, revealing yet another link to gelatinous filter feeders. In the literature, the diets of shallow-dwelling species, characterized or analyzed through visual gut content analysis, are dominated by small crustaceans and characterized by the absence of gelatinous prey. On the other hand, the diets of deep-dwelling siphonophores, assessed primarily from ROV observations, are dominated by large prey like shrimp, fish, squid, jellyfish. But when we include these metabarcoding results to the picture, we find that gelatinous and soft-bodied prey are just as prevalent in shallow habitats as they are in the deep ones. And similarly, on the other hand, the small prey that is expected to be missed by the ROVs appear to be just as prevalent in the deep sea as it is in the shallows. So in conclusion, using a unified methodological framework, I found novel prey types that were undiscovered by visual methods, especially hidden links to gelatinous animals and filter feeders near the base of the food chain. These results confirmed or supported some of the morphological predictions for several species, further supporting this causal link or relation between Tentilla and diet. And finally, I found that the apparent trophic differences between deep and shallow siphonophores as a group are just artifacts of the confounding methodological biases. These last two chapters have addressed the trophic role of siphonophore diversity, indicating that different species are playing different roles across depth habitats. And they do so by building Tentilla specialized to strongly select for different prey types. Before I'm done, I'd like to give you all a quick glimpse of the work I am currently carrying out at the Southern Land Lab here at UO. I'm now working on salps, another group of marine gelatinous animals. Salps feed by filtering phytoplankton and bacteria, and also form colonies of many clonal bodies, some of them also meters and meters long. Different salp species present a variety of colony architectures with different relative angles and axes to the direction of swimming. And these are hypothesized to relate to swimming speed and efficiency in response to different selective pressures. And I'm setting out to tackle this question from a phylogenetic approach. So looking at the evolutionary history of colony architectures using an 18S gene phylogeny built, it seems that they all derived from a fast swimming linear chain ancestor into all of the strange diversity that we see today. From a phylogenetic perspective, swimming speed is conserved, phylogenetically conserved, and it drops very low in those clades that have lost the linear architecture as they become much less hydrodynamic. To connect these morphologies back to the efficiency of the swimming and their energetic trade-offs, I'm collecting different subspecies underwater of Hawaii as soon as Kelly said, a week from starting. And I do so instead of using regular jars, I've been using these airtight containers that have installed optodes inside. Thus, when I took the salps out of the water onto the boat, I was able to measure their oxygen consumption rates as they continuously swim around in the jar. Oxygen consumption per body mass was consistently higher in those species with faster swimming speeds, such as the linear chains here in orange. And this may seem trivial, but bear with me, because the energetic efficiency of swimming in these linear chains, despite having a more streamlined shape and a more efficient multi-jet propulsion arrangement, is worse than the other species. And in Kelly's words, it's like comparing a Ferrari with a Prius, speed versus efficiency, energetic efficiency in this case. And this suggests that speed might be related to other functions, such as escaping predation. And that's kind of where we're at right now. This is all I have put together for you today. I would like to thank all of the funding sources that have made all this research and my trajectory possible. And also all the people who have contributed to this research in one way or another, and those who have guided me through this hard journey. And thank you all for your attention, and I'll be happy to answer any questions you have.
Peter, would you like me to handle the questions?
Yeah, go for what did you say?
Do you want me to handle the questions?
Yeah, go for it.
Okay.
Check the chat.
So, okay, it looks like there's a question in the chat, so maybe we'll start with that one. So let's see, gorgeous presentation and animation, so many fascinating videos. I agree there. This is from Monica, by the way. Let's see, do siphonophores have any predators, or are their nematocysts and Tentilla enough to deter predators? So that's the first question.
Yeah, that's a great question. Actually, yes, siphonophores have many predators. Let me see if I can bring up one of those first slides here. I don't know if you can see this. So the color of those lines that connect all these different animals in the midwater represents the color of the predator. So you can see that those green circles that have all of these things that siphonophores eat, they also have lines from other colors that lead to them. All of those are predators of siphonophores, and that includes a bunch of fishes. It includes also a bunch of jellyfish, like there's some called narcomedusae that like catch all sorts of other gelatinous animals as prey, including siphonophores. There's some observations that are a bit dubious, but they indicate that some siphonophores eat other siphonophores. As you can see, a little loop-de-loop up there. And there's some super specialized siphonophore predators. Like there's these tiny deep sea slugs called cephalopyges that not only they're not deterred by the Tentilla, they love them. They love Tentilla. They will like munch on the tentacle, like, I don't know, like a bit like a Pac-Man cartoon, eating all until they eat the whole tentacle, and then they keep going to the next one. And there's these amphipods that will carve out different bodies. They will live inside and eat them alive, or just pluck out their bodies and use them as flotation and eating. Slippery lobster larvae, siphonophores, all sorts of things. There's a broad variety of predators, yes. Oh, and sorry, what you said about defense. They do have defensive nematocysts on their bodies. They do have these bracts that are like protective shields, and they're packed with defensive patches. And those nematocysts are completely different from the ones in Tentilla. Those are optimized for defense and not for prey capture. Yeah.
Looks like Susie has a question. Maybe we can circle back to Monica's second question. Go ahead, Susie.
Oh, okay. Yeah, that was a really awesome talk. Super fun. Haven't seen anything like that in a long time. But I was wondering what's known about what governs whether nematocysts will fire. So I'm thinking about the specialists versus generalists, their chemosensory cues, movement of the prey. Is there anything known about that?
Yeah, I mean, there's general understanding of how nematocysts work in general, or at least in hydrozoans, which means that there is chemical conditioning, which means the main trigger is always mechanical. Triggering is mechanical, but mechanical triggering will only happen given the conditioning of certain chemoreceptors on the cell. Nematocysts are homologous in the same way as like retina cells. They're all homologous to neurons. These are modified neurons. They're super sensitive and like they have very complex electrochemistry and stuff. In the case of siphonophores, so the Tentillum discharge elicits nematocyst discharge, but the Tentillum discharge has its own mechanism for firing. So once the Tentillum fires, all the nematocysts fire. There's no one without the other. But for a Tentillum to fire, for crustacean specialists, like there's been a few studies that like it's the tugging. So just imagine a grenade ring. When you pull the grenade ring, the grenade fires. So when you pull on the terminal filament, that is like a glue line, because they're trying to escape because they got entangled, then that fires, and then the nitoband, the one with the nematocyst, shoots, and it actually like spans several centimeters and aims to the target, and that involucrum, that kind of like little membrane, is kind of like the barrel of a gun, and it aims the sling, and then it wraps and then it pulls. But yeah, what I've noticed in my own experiments trying to elicit all this discharge is that the big ones, the bigger Tentilla you have, the harder it is to make them fire on command. They're just like much pickier. They're like, I spent a lot of resources building this. You're not going to trick me into firing this. They could like bump into like containers and whatever, and they're not going to fire. And they're like, okay, now we're going to fire because this is expensive. But still haven't figured out exactly the triggers.
So that could be potentially an interesting area of your phylogenetic analysis, to study the chemosensory conditioning in a phylogenetic framework for the generalists and specialists, maybe.
Yeah, that would be cool. I just have no idea how to do that. Thanks.
So see, part two of Monica's question was, is there any effect of climate change or changes in temperature on siphonophores? So have populations increased or decreased? And then she notes, in the past decades, I've heard a lot more news about Portuguese man-o-war washed up in beaches in my country, Indonesia. Not sure if their population is actually increasing or just more people are reporting those interactions.
Yeah, so one reason we don't have general understanding on how siphonophores as a group react to climate is that there's no fossil record. So we can't like do a paleontological connection between that to get a general sense of what to expect. Also, like the phylogeny can be dated without a fossil record, so we also don't know what a lot of these transitions happen. But this is not, I know this is not your question. The thing is that a lot of the finding the Portuguese man-o-war in different places have been changes in the currents and the winds that some of them are due to climate changes. More generally, people have studied changes in the proportion of gelatinous predators in these marine ecosystems as a response to decreased competition with fishes and fisheries, things that are not directly climate change. They can be, but other anthropogenic impacts. The thing is that there's only like, I think, one time series on, other than the Portuguese man-o-war, on another siphonophore, and it's on Nanomia in only one location in the Monterey Bay. And there's changes that are associated with El Nino and stuff, but general trends to having more siphonophores in the future or less siphonophore in the future are completely unknown right now.
Peter, did you have a question?
I have a question. How closely related, like how many million years are the phylogenetic trees you were showing us?
We have no idea. Yay! We can apply a general molecular clock, but we know that in cnidarians that can vary a lot with the group. So when you don't have any fossil to ground that time, we're talking about like, you know, tens to hundreds of millions of years of uncertainty in using a molecular clock alone. There's some things that's something that I have spoken with more molecular phylogeneticists about, and that we could just integrate across several groups and try to figure that out as a next follow-up project, especially because the fishes that siphonophores eat are all actinopterygians, which appear which diversified after the KT boundary, with a lot of changes in, you know, the oceanography and like the biodiversity of the oceans. And it would be really interesting to see the rise of siphonophore fish specialists with the you know, burgeoning of fishes as an abundance of actinopterygian fishes in the ocean and their larvae and all of that. But yeah, sorry, I don't know the answer to the question.
Great. Other questions? All right. Shall we end there, Peter?
Yeah, and oh, right. Are you all planning to hang out on longer for tea time or not?
Yeah, so what's the timing of tea time again, Peter?
People sometimes stay on the come back to the Zoom call to chat more. Sorry, I forgot to ask this at the start of the talk, but if people are interested, are you all interested in sticking around for 20 minutes?
I'm happy to stick around.
Okay. Anybody?
I can stay for like another 10 minutes or so. But
okay.
Yeah.
All right. Well, yeah, that was great. Let's thank Alejandro again.
Thanks, Alejandro.
Thank you.
Very cool talk. So I guess we stay on this link. I don't think I've done the tea time since things have been virtual, but I guess we just stay here and people get tea and then come back to the same Zoom link.
All right.
So you should feel free to get up and take a little breather if you want to.
Yeah. Thanks. Yeah, that would be good.
Yeah. I had sort of forgotten how the tea works, Peter. I don't think I've been to a tea since.
Yeah, I forgot to. This is no longer being recorded by. Wait. There. Now it's really no longer being recorded.